The Agrobacterium-mediated transformation is an easy method for producing genetically modified potato plants in order to understand specific gene functions and their contribution to organ physiology.
Transformation by Agrobacterium rhizogenes is a robust tool for rapidly assessing gene function in roots, as similar results can be obtained by Agrobacterium tumefaciens transformation.
Demonstrating this procedure will be Sandra Fernandez-Piñan, a postdoc, and Jennifer Lopez, a master's student from our lab.
Begin by growing a single Agrobacterium colony overnight in 5 milliliters of yeast extract broth, or YEB medium, supplemented with antibiotics in a 50 milliliter centrifuge tube at 28 degrees Celsius and 200 rotations per minute. For a tumefaciens transformation, measure the optical density the next morning. When the optical density at 600 nanometers, or OD600, reaches 0.6 to 1, centrifuge 1 milliliter of the Agrobacterium culture and resuspend the pellet in 1 milliliter of fresh YEB medium without antibiotics. After the second wash, resuspend the pellet in fresh YEB medium without antibiotics to the appropriate concentration to obtain a final OD600 of 0.8 and place the bacterial cells on ice. For plant transformation using A. rhizogenes, carefully transfer a donor plant from a 2 ms medium culture to a 120 by 100